In this video, we're going to briefly talk about the different types of interactions that our various side chain classes can perform. And this is all part of the tertiary folding. So remember, when a protein folds and the amino acid side chains are interacting to cause that protein to fold, that is the tertiary structure. The amino acids are interacting together, and they can interact together by 5 different types of bonding. We have the van der Waals interactions, we have the hydrophobic effects, we have hydrogen bonds, ionic bonds, and disulfide bridges. I'm going to start with the van der Waals interactions. The van der Waals interactions are fairly simple. This is just basically the magnetic attraction that all things have together. They basically have everything that has some mass, even if it's a tiny bit of mass like the atomic level we're talking about here. Everything with mass is going to have some gravitational pull. And so as different atoms get close together, they're going to have that pull together in a little bit of interaction. This is extremely weak, but over the course of thousands and thousands of atoms interacting together, they can add up. Basically, when you think of van der Waals interactions, every one of our amino acids can do this, all 20. So nonpolar, polar, uncharged, and then our acidic and our basic with their charges. All of them can do van der Waals interactions. The hydrophobic effects are basically when our nonpolar amino acids congregate together. So remember, polar things like to interact with other polar things because of hydrogen bonds, and nonpolar things interact with other nonpolar things. This is why water and oil separate, and the oil clusters together, they're all nonpolar. So those nonpolar amino acids, so basically everything in this grouping up here that has all of the side chains that are hydrocarbons, these are all nonpolar side chains shown here in blue, they're all going to interact well together. And importantly, they tend to aggregate towards the center of the protein when it starts to fold, because remember, the protein is in an aqueous solution. Our cells are in aqueous solutions. And so they're all inside of water, and those nonpolar amino acids want to congregate together in the middle of a folded protein in order to get away from the water on the edges. So the hydrophobic effects tend to pull those nonpolar amino acids together towards the center of a folded protein, and pretty much only the nonpolar amino acids are going to undergo hydrophobic effects. Only nonpolar likes to interact with other nonpolar. Hydrogen bonds, on the other hand, require polarity. They require opposite dipoles to attract. And so anything with polar bonds, and that includes our polar uncharged amino acids, all these have polar bonds in here. That includes our polar charged, our acidic and our basic as well. Any of those types of amino acids can create a hydrogen bond. For example, if we take a look at asparagine here, this carbon double bond oxygen could make a polar bond. That oxygen would have a negative dipole. It could make a polar bond with the hydrogen over here of a glutamine that has a positive dipole. And we can see that over here in these hydrogen bonds right here. So this would be a hydrogen bond. That hydrogen would have a positive dipole because it's bound to that oxygen, and this oxygen bound to this carbon would have a negative dipole. There's a hydrogen bond holding those two different amino acids together. So in order to be able to do a hydrogen bond, the amino acid side chain would have to be polar or charged. Ionic bonds are when you have complete charges interacting, and so the only ones that can do that are those amino acids that have a complete charge, and so now we're limited to acidic and basic. And it is important to remember that opposite charges attract. So if you have a basic amino acid, it's going to interact with an acidic one. The positive charge interacts with the negative charge. And so we can see that up here. We have an acidic amino acid with its negative charge interacting with a positive amino acid. And it doesn't even just have to be other amino acids. They can interact with anything with the opposite charge for an ionic bond. And then finally, the unique one is a disulfide bridge. And what you have to remember here is only cysteine can do a disulfide bridge. This is when the sulfur in that amino acid side chain actually creates a covalent bond with another cysteine. So here we have a cysteine over here, another cysteine a long ways away in that primary structure, but they can form a disulfide bridge, and that will help this protein fold. So these are the various types of interactions that the amino acids can do for both tertiary, and then these same side chains can do the same interactions for quaternary structure if it's bound to another protein as opposed to within the same protein. Now remember, the way a protein folds is really important to what its function is going to be. So if you change the fold, you change the function. And this is the whole idea of denaturation. anything that denatures a protein interrupts these interactions. They can interrupt the hydrogen bonds, the ionic bonds, and they can do that by heat or by altering the pH. But by denaturing the protein, you're breaking these interactions, you're unfolding the protein, and therefore you're losing its structure and losing its function. But remember, that denaturation only happens down to the level of primary structure. It does not break the covalent bonds holding the amino acids together in the primary structure, only the interactions that help the folding.